Hi guys, let's start with our next chapter, chapter number 16, chromosomes, genes and proteins. The overview of chapter, it comprises of understanding of chromosomes and genes, learn genetic inheritance patterns, explore cell division processes such as mitosis and meiosis, and comprehend how genes determine protein production. We will start with the allele transmission mechanics. So, gamete formation, key characteristics are gametes contain half the chromosomal complement. Human gamete 23 chromosomes versus 46 in the body cells. So, these are 23 pairs of chromosomes. So, individually, if you will see, it will be 46. Each gamete carries one allele per gene pair. Alright, now let's talk about genetic diagram. Monohybrid cross methodology. Steps for creating genetic diagrams: you can record parental phenotypes, you can record their genotypes as well, and then identify the possible gamete combinations. Predict offspring's genotypes and phenotypes, and then you can also calculate the probability ratios. For example, here we have inheritance probability table. We have capital G and we have small g. Capital G is linked with gray and the small g is linked with charcoal. So in the genotype we have three types of combination. If it is both GG, capital GG, so the phenotype is gray and the probability will be 25%. If it is one capital G and one small g, then it will be gray and the probability will be 50%. And if both are small g, then it will be charcoal and the probability is 25%. Like this you can make the combinations, okay, of parents' genotypes and phenotypes as is mentioned earlier, and you can also predict the probabilities. Okay, some genetic terminologies like genotype, genetic composition, phenotype, observable characteristics. So you can say that these are the physical characteristics. So if they are physical characteristics, called as phenotype, and if there is genetic composition, genes, then it will be the genotype. Okay, allele means alternative gene form. Hetero means different. Heterozygous means different allele pairs, and homozygous means identical allele pairs. Now let's talk about meiosis visualization. Now genetic inheritance principle. Okay, here we have probability calculations. Equal chance of inheriting maternal paternal alleles, maternal from mom side and paternal from father side. Predictable inheritance patterns. Advanced genetic concepts include codominance. Example such as multiple alleles can be expressed simultaneously. Blood group inheritance demonstrates complex genetic interactions. In genetics, inheritance and phenotypes, we can discuss dominant and recessive alleles, pedigree analysis. Now key concept here is dominant alleles masks the effect of recessive alleles. So these are the dominant one and these are the hidden one, recessive ones. Notation: dominant allele typically represented by uppercase letter and recessive allele represented by lowercase letter like small r. Guinea pig hair coat example. Inheritance pattern. Now rosette hair capital R is dominant over smooth hair small r. So genotype determination methods. In such case, how will you determine genotypes? So you analyze offspring phenotypes first. Consider possible parental genotypes. Use genetic diagrams to predict inheritance. Like a table we did before, Punnett table, you can make a table like that and you can predict the probabilities, and you can also make the genetic diagrams. Now let's talk about test crosses. Purpose is determine unknown genotype of an organism with dominant phenotype. Procedure is cross suspected heterozygous organisms with homozygous recessives. Then analyze offspring phenotypes. Your interpretation can be presence of recessive phenotype suggests heterozygous parent, Absence of recessive phenotype suggests homozygous dominant parent. Now let's talk about sex determination and sex-linked characteristics. Chromosome-based sex determination, female chromosomes we represent it as capital XX, and male chromosome we represent it as capital X and capital Y. Inheritance probability is 1:1, like 50-50, chance of male or female offspring. Sex-linked genes located on X or Y chromosomes, unique inheritance pattern, example red, green color blindness. Now let's talk about color blindness genotypes. Genotypes capital X, capital B, capital X, capital B. So it's female, all capitals, and the phenotypes you can say that normal vision, for example. Another is capital X, capital B, capital X, and small b. Here also female. However, the phenotype can be carrier, that she is the carrier of some genetic variation. One is capital X, small b, small x, small b. Now here also female, but maybe some genetic disease such as color blindness codes are transmitted from parent to the offspring. Similarly, another genotype capital X, capital B, capital Y, it could be male but with the normal vision. Capital X, small b and capital Y, again male but with a genetic linked problem such as color blindness. Now inheritance of sex linked traits, more common in male due to single X chromosome. Carrier females can pass recessive alleles to offspring. Key genetic principles. First is pure breeding. Organisms consistently produce offspring with identical characteristics, always homozygous for specific traits. Genetic diagram can help you interpret, such as you can use Punnett square methods, track allele transmission across generations, predict offspring phenotype and genotype. Some practical implications and examples of genetic inheritance includes wing types in fruit flies, hair color inheritance, blood type inheritance. Gene and protein synthesis. Here we will talk first about DNA structure and composition. DNA molecule basis is composed of two long strands of nucleotides. It has 4 different bases, and these are capital A, capital C, capital T and capital G. Bases form a double helix structure, like DNA structure is double helix like this. Okay, base pairing follows a specific rule, like A will pair with capital T and capital C will pair with capital G. Now protein molecules fundamentals. Remember, these points are very important. They are made of long chains of amino acids. Secondly, they have 20 different amino acids, okay, that exist. Then we have amino acid sequence that determines protein function, determines protein shape. Shape affects protein specific role. Example, amylase enzyme shape determines its ability to break down starch. So the important points are long chain of amino acids. What we are talking about the protein molecules. So there are long chains of amino acid, there are 20 different amino acids exist, and the sequence determines the protein function. The amino acid protein shape determine a specific role, such as an example is given here, that ability to break down the starch. Alright, now let's talk about gene and protein relationship. Gene definition. They are length of DNA coding for a specific protein, number 1. Number 2, base sequence determines amino acid sequence, remember. And then number 3, that it controls cellular functions that determines protein shape. Now let's talk about protein synthesis pathway and key protein synthesis steps in this. So first we have mRNA. Okay, it carries genetic information from DNA. After that, ribosomes links amino acid in a specific order. After that, requires presence of all 20 amino acid that we just spoke earlier about, and then protein shape determines its function. Okay, cellular specialization, gene expression. Every cell contains same genetic information. Not all genes are active in every cell. Specialized cells express only necessary genes. Skin cells like keratin production, nerve cells like neurotransmitter receptor proteins. Next we have stem cells, unspecialized, undifferentiated cells. Can divide and become specialized, found in embryos and adult tissues. Potential medical applications, regenerating heart muscles and producing nerve cells. Next we have genetic inheritance concepts, chromosome characteristics, haploid cells 23 chromosomes, diploid 6 46 chromosomes. However, they are in the pairs of 23. Homologous chromosomes contain similar genetic information. Now inheritance mechanism, we have mitosis here, identical genetic reproduction in mitosis. If meiosis, genetic variation through cell division. Allele inheritance determines genetic traits. So this is three inheritance mechanism. Now some research and future potential such as stem cell research is going on. It's an emerging field with significant medical potential. Ongoing discoveries about cellular differentiation is going on. Promising treatments for previously untreatable conditions. So our key takeaways, DNA controls cellular function through protein synthesis. Genetic information is consistent across body cells. Specialized cells activate only necessary genes, and stem cells offer revolutionary medical possibilities. Alright, now let's talk about chromosomes, mitosis and meiosis. Okay, let's talk about chromosomes and cells basics first. So in chromosomes fundamentals, we will talk about chromosomes composition. Humans have 46 chromosomes in each cell, arranged in 23 pairs, one set from mother and one from father. Begins with zygote formed by fusion of egg and sperm cells. Okay. Now for the types of chromosomes, we have number 1 haploid cells and number 2 diploid cells. Haploid cells contain a single set of chromosome, example gametes, egg and sperm cells. Diploid cells contain 2 complete sets of chromosomes. Okay, example zygote after egg and sperm fusion. Important points, try to remember these as it can be tested in the exams. Chromosome characteristics, homologous chromosomes, paired chromosomes with similar genetic information, one chromosomes from mother and one from father, matched in size and genetic content. Next we have mitosis, cell division. Key characteristics of mitosis. Purpose is produces genetically identical cells used for organism growth, body repair, asexual reproduction. Chromosomes preparation. Chromosomes are copied before division. Each chromosome consists of 2 identical chromatids. Centromere joins the 2 chromatids. Okay, so this is how the chromosomes prepare itself. So first they are copied before division, then each one consists of 2 identical chromatids, then centromere joins the 2 chromatid. Okay, now let's talk about mitosis process. Here important mitosis points are: it produces daughter cells with identical genetic material. It is critical for healing. Example skill cell regeneration. Used in plant reproduction, example potato plant and stem tubers. Alright, now let's talk about meiosis. Meiosis fundamentals, purpose, create gametes with half the chromosome number, enables sexual reproduction, reduction, division. Diploid parent cell produces haploid daughter cells. So it reduces from diploid, it becomes haploid. From 2 to it become 1. Chromosome number is halved. In humans, 46 chromosomes are there, which comprises of 23 pairs of chromosomes. Now in meiosis process, we have first division, meiosis 1. Here homologous chromosomes pair together. Chromosomes separate randomly. One chromosome from each pair goes to the daughter cells. For now, this much is important. More we can discuss when we can see the questions, and then accordingly we'll make our videos. Okay, for now let's talk about genetics and inheritance. Here we can understand genotype and phenotype. Genotype and phenotype basics. Okay. Alright. So definition first. Genotype, the genetic makeup. Okay. Genes. So genotype, the genetic makeup of an organism, specifically the genes an organism possess. Next is phenotype, the observable characteristic of an organism, including physical trait like fur color, invisible characteristics like protein composition in cell membrane. Now chinchilla fur example here. Here we have color example, GG light gray, fur, capital G small g light gray again, small g charcoal. So this is 25%. We remembered that we have done before. Okay, this is also 25%. Okay, like this, and this is remaining is 50%. Okay, now dominant and recessive alleles. Key characteristics: dominant alleles G expresses its trait even with only one copy, mask the effect of the recessive allele, produce the same phenotype whether homozygous or heterozygous. Recessive allele small g only express its traits when two copies are present, requires homozygous recessive conditions to show phenotype. In heterozygous state it remains hidden. Okay, so hidden is recessive allele. Remember these points, guys, for the exam perspective. Carrier concept is a heterozygous organism which is G and G, capital G and small g. It carries the recessive alleles but does not express its phenotype. So potential to pass the recessive trait to offspring. Now codominance, what is it? The definition is neither allele is completely dominant or recessive. We were talking about that one gene phenotype is dominant and it makes the other recessive, but here in codominance, all right, neither of them is completely dominant or recessive. Both alleles contribute to the final phenotype. Example, flower color inheritance. Notation rule: use superscripts to distinguish codominant alleles. Avoid using capital and lower case letters to prevent dominance confusion. Okay. Alright, so you can practice like this, making tables. Now we have blood group inheritance example. A, B, O, blood group antigens. Gene represented by capital letters, capital I. Three possible alleles, IA, IB and I with a small naught, small 0 on top. IA and IB are codominant. Both IA and IB are dominant to this one, I with the small knot on its head. Genotype, phenotype, blood group. So if it is capital I, capital A, capital T, capital A, the phenotype, the group will be blood group A. Okay, and the blood group A. Similarly, if it is genotype IAB, blood group will be AB. Okay. If it is capital I and capital B, again capital I, capital B, so the blood group will be B. And then one knot, another one knot like small O on top, so it will be blood group O. Okay. With this we have finished our this chapter. See you in our next chapter.